Once RNA normalization and defining cell types is completed, the data can be explored in many ways within MAS. In addition, single-cell data with the cell type annotations and quantified data can be exported to a variety of file formats or live streamed to other applications. We will start here in the Analysis tab. You can create a new analysis dashboard by clicking on the plus sign here. Provide a name and choose the type of analysis that you're conducting. Here we have two choices, cell type and cross-sample. Later in the video, we will discuss cross-sample analysis. For now, we will choose the cell type analysis, using which you can explore the differences between different types of cells using CITE-Seq or UMAP projections and additional differential analysis provided in the dashboard. Choose the projection that you would like to display that this dashboard should use. In this case, we will use the CITE-Seq that was created earlier. The dashboard provides a comprehensive view of all aspects of the dataset. At the top here are different samples, and you can choose which sample to display by simply clicking on one of the rows. Then you can highlight cells of different types by clicking on any of the cell types in the side panel. In addition, you can search for specific markers in the side panel here, and RNA or ADT markers, and overlay the UMAP or CITE-Seq plot with that marker in a pseudo color format, colored by the marker expression. In addition, you can choose specific cells of interest within the plot and understand how these cells differ from the rest of the cells by looking at the differential expression table here. Finally, you can also conduct some differential analysis between different cell types, for example, by choosing a specific cell type versus the other cell type that one wants to conduct the differential analysis against. The results are displayed either as a volcano plot with the x-axis showing you the log-fold differences between the cell types and the y-axis showing the negative log 10 of the p-value, meaning higher the value here, the lower the p-value or generally of interest here. The results are also available as a table, and all of these can be downloaded by clicking on the download button here and choosing what aspects of the dashboard should be exported. Finally, if there are specific plots of interest, you can click the pin button here, which will essentially create a carousel of plots that are of interest. For example, this plot is for this sample. We can change the sample using the same marker and click on this pin button to get another of these plots saved. These plots can then be exported as high-resolution images, and they're automatically included in the export package when you export these results. A different type of analysis is to compare samples to each other by protein and RNA expression in defined cell types across the samples. Here we are expecting that your samples have been annotated with information that can be used as dependent variables to compare protein and RNA data. MAS allows you to perform different types of analysis based on the type of dependent variables. Grouped analysis, when the samples are categorized into multiple groups by a dependent variable, for example, samples from different response and treatment groups, or alternately, samples may be compared on a paired basis. For example, for each subject, you may have samples from before and after treatment. Finally, the dependent variable may be a continuous instead of being categorical. That is also possible conduct using MAS. For example, in this case, we have 6 samples here, and pairs of samples here belong to different donors, 3 donors, And each of these have been treated with have been subject to different ex vivo treatments. And here we are trying to understand which RNA or protein markers are different and which cell types across these treatment options. As such, we have chosen the dependent variable to be the treatment. In this case, the samples are not paired as pre or post treatment or any other way of pairing across the subjects. So we are not going to choose anything for pairing. Finally, you can also conduct this analysis in subgroups of the samples. In this case, we do not have any major subgroups, so we are not going to select it, but you can imagine if you had donors or subjects categorized into, for example, by gender, male, female, and then you can conduct the subgroup analysis within each subgroup, and mass will present the statistics within each subgroup. You can also set a DE threshold, meaning how much variance should you expect across your samples, or differential expression do you expect between different groups at which you're interested in, above which you're interested in looking at the results. So now that we have chosen this configuration for our analysis, I can hit run. What MAS is doing right now is conducting for each RNA ADT dimension in each cell type, comparing between these treatment groups and bring us back statistics for that comparisons. So what you're seeing here are, on the left-hand side is the familiar cell type tree. The plot in the middle here, in this case, because we have multiple groups, or 3 groups more precisely, the analysis that was conducted is one-way ANOVA analysis, and the x-axis is showing you the standard deviation or the variance, square root of the variance across all specimens, all samples. And on the y-axis is the negative log 10 of p-value, meaning higher the value, it is of more interest to us. This is similar in many respects to a typical volcano plot. If we had only 2 groups instead of 3 groups, the x-axis would have been the differential expression between the groups, and the y-axis would continue to be the p-value. And the points of interest in this case, for example, are those that have high variance, meaning they have reasonably large expression across the different samples, and have a low p-value, meaning the negative log 10 of the p-value is high. You can also focus on specific cell types by clicking on any of these cell types here. And then, for example, I can click on the CD4 positive T cells, and you can see which markers within the CD4 positive T cells are interesting. We can further examine each of these data points. So this is each point here corresponding to one RNA or one ADT. One ADT dimension. In this case, I'm looking at the expression of CD69, and it shows that this marker is differentially expressed in the samples treated with CSC, while not expressed in samples treated with PHA or TNF-alpha. You can further drill down by clicking on these individual points and examining how the single cell data looks across these for the CD4 positive T cells between the samples that have been treated or not treated with the respective treatments. In addition to the analysis we discussed, MAS provides multiple mechanisms to export either plots or data. Under the Data Extract tab, there are 2 sub-tabs, Reporting and Data Extract. Under the Reporting tab, one can create overlays of single-cell data across different configurations of samples, markers, and cell types. So for example, here, we can select different markers of interest. Select which cell types we would like to display. For instance, I'm going to choose T cells and B cells here, and then configure whether we would like to see multiple samples, markers, or cell types on each plot. For example, when I have samples selected, what you should see is for B cells, for example, for this marker, how the expression looks across the different samples. We can toggle this, for example, to cell types. And now what you see is the T cell and B cell expression within each sample for that marker. You can export these plots by simply clicking on the download button here, and it will export to a PowerPoint slide deck. In addition, an automatic threshold for positivity is computed, and you can adjust this threshold by simply clicking on any of the plots and hitting save to adjust the threshold. Optionally, you can choose not to show the threshold. If the threshold is chosen, positivity for each of the cell type for each marker and each sample is computed and displayed as a table, which can also be exported to an Excel file. Finally, if you have specific configurations of these overlay plots that you would like to save for later use, you can click on the save options. You can give it a useful description. Save as a new query, and then when you return to this page later on, you can simply click on this to have all the options pre-filled, and you simply have to click on submit to recreate the plots that were previously created. The last tab here is data extract, which you can use to export the data for a data set to various file formats. There are a couple of file formats here of high interest to us. One is H5AD or CSV formats. For both of those formats, you can export single cell data that includes the expression levels for every cell for ADT, RNA, normalized RNA, along with cell type annotations and any projection dimensions that have been created. A second format that is of high interest is the metric data. What this exports is either the median marker or marker values, count values, for each cell type, both for ADT and RNA, or percentages of cells relative to their respective parents. For example, T cells as a fraction of lymphocytes. Both of these formats are provided as an OData link. If you click on this OData link, what you get is a link is copied to your clipboard, and you can paste this link into many applications, many downstream applications like Excel, Tableau, Spotfire, or a Python script or an R script to create a data frame of the median value data and then conduct downstream analysis for the other purposes.